Hi, my name is Pierce Alonso, and today I'm going to be talking a little bit about CRISPR-Cas9. We're going to be talking about the future of genetic engineering and really humanity. This is for Biology 107, and this is one of my honors presentations. Before we get into it, this presentation is going to be split into 6 categories. First is the history and function. We're going to be moving on to disease, the idea of designer babies, how CRISPR could be a fountain of youth, the ethics of CRISPR, and setting realistic expectations. Each category will be denoted with their symbol in the corner, as such. So what is CRISPR-Cas9? Well, it's actually an acronym, and it stands for Clustered Regularly Interspaced Short Palindromic Repeats. Now, this is a very confusing acronym, but it's not really that important. What is important, though, is the function and application of CRISPR. Cas is just any CRISPR-related proteins, and Cas9 specifically is just the first protein that we learned how to program. Okay, so let's get into what is CRISPR. Since the dawn of time, viruses and bacteria have been at war. We're going to talk a little bit more about this in a second, but CRISPR and Cas9 occur naturally in many bacteria as a defense mechanism against viruses. The way it works is it saves the DNA and uses it to kill that virus later. So what does that mean? Well, so here we have a bacteriophage. It's a type of virus, but many viruses work in the same way. So they latch on to a bacteria, and then they inject a string of DNA into it. Well, most of the times, bacteria do not survive this process. After the virus has run its course, the bacteria will be dead. But there are certain times when bacteria actually do survive this. When they do, their CRISPR Cas9 proteins will save a little bit of the DNA that they found from the virus, a little bit of the DNA that they injected into it. And they save a little copy in the form of RNA. And as we know, bacteria don't have a nucleus, they have nucleated regions. And these little Cas proteins will ride through all of the DNA in the bacteria, and they look for anything that matches the DNA from the virus. If they find it, they cut it and they kill that DNA. Cut it right at the point and it dies. But why is this important? I mean, it's cool and all. It seems like something of science fiction. So why is it so important? They're programmable. So scientists have found out that they could program these proteins and have them cut anywhere. Remember those little RNA strands? Yeah, we can give Cas proteins those little RNA strands and have them kill any DNA anywhere. And better yet, we have found Cas proteins that are not only able to cut out DNA, but they're able to insert other DNA into that same spot. So now let's talk about some applications. So what does this mean for disease? I'm going to be talking about a couple diseases here that have shown some promise for being cured or helped with the help of CRISPR. So first, let's talk about blindness. So many hereditary forms of blindness are caused by specific mutation, which makes it easy for CRISPR to target and modify that single gene, effectively eradicating that disease. Not all forms of hereditary blindness, but many forms are caused by that one specific mutation. Now let's talk about AIDS. While CRISPR does offer several ways to potentially fight against AIDS, one way is by programming Cas proteins to cut the DNA of the HIV virus, killing it where it hides in white blood cells, rendering the virus completely useless. Studies have already started in China, and actually just a few months ago, the first person was cured of AIDS using CRISPR. It has not been replicated, so we don't know if it is going to be very common going into the future, but it has happened. Now let's talk about muscular dystrophy. Most forms of muscular dystrophy are caused by specific mutations in the DMD gene. Research in mice has shown that it actually may be possible to correct this mutation and reverse the effects of muscular dystrophy. And with Down syndrome, studies from UCSD have shown that it may actually be possible to cure Down syndrome or at least stop the progression. They would do this by targeting the third chromosome 17 in several locations and effectively disabling the entire chromosome. Also, we got to be realistic. This isn't going to cure every single sickness. As we know, diseases mutate over time, and there are many diseases that just aren't genetic. but we may be able to use CRISPR to develop stronger immunities to these diseases. Now, one topic that keeps bringing up all the time when talking about CRISPR is the idea of designer babies. Some people think of having designer kids that are genetically designed to be super strong, super intelligent, or have specific characteristics such as a round nose or blue eyes. But the fact of the matter is that we're just not there yet. The science isn't there. We don't even know all of the genes that contribute to something as simple as blue eyes or high intelligence. But that doesn't mean that designer babies don't exist. Meet Cosmo. Researchers at UC Davis were able to insert an SRY sex-linked gene into chromosome 17. They chose chromosome 17 because they found a place where the gene would be expressed safely. The modified gene causes Cosmo's offspring to have a 70 to 90% chance of being male, as opposed to the natural 50%. Now, I think that this proves that designer babies do have real-life current applications, and I also think it proves that all of the stereotypes of UC Davis are warranted. Now let's talk about the eternal youth. Many people think that having eternal youth from the use of CRISPR is possible, and it may be possible, but the reality is that we don't actually know. We don't know how much of aging can be attributed to genetics, but we do know some organisms, such as lobsters, are immune to aging, and we may be able to use CRISPR to learn more about the genes of these lobsters and maybe apply that to humans. One of the biggest problems with CRISPR is the ethical concerns, and this is something we need to talk about. One of the biggest things that gets brought up is somatic modifications versus germline modifications. Now, somatic cells, as we know, are the cells in our body that do not contribute to our offspring. Now, somatic cells and modifications with somatic cells would be fine. It would be your own choice. But germline modifications means that those modifications would be passed down to your offspring. And this brings up the issue of consent. How do we know that the kids that we have want to be super intelligent, or that they want us to choose that they have blue or green eyes? We can't. So that's one of the biggest ethical concerns with it. And also the responsibility. If a child is born with a huge deformation caused by CRISPR, who is responsible? Is it the medical professionals? Is it the parents? Is no one responsible? It's still yet to be decided. Another concern that gets brought up is what is stopping a country like North Korea from using CRISPR to inflict the rain and make an army of super soldiers that are designed specifically to be incredible soldiers? We don't know, and we can't stop anything like this from happening. So these are things that we need to think about going forwards. And lastly, let's talk about a realistic future. Really, CRISPR starts with tackling diseases. Bodily modifications like breathing underwater and some crazy ideas that have come out, that is probably very distant. Although we can't take it off of the table just yet, we can't rule out the possibilities, but that is just very far into the future. Cost is very prohibitive at the moment, as even just small CRISPR modifications cost insane amounts of money, but we can look into a bright future and knowing that in just 5 years, the cost has fallen over 100 times for CRISPR modifications. So maybe very recently, or maybe very soon, it will actually be realistic. So that is my presentation. I really hope you guys enjoyed. I hope you guys learned something and you're able to take something away from this presentation. I hope you guys are excited about this new science as I am, and thank you for watching.